Next topic is cell-to-cell signaling. So we have four cell-to-cell signaling processes that I want to go over. The first is the gap junction. Gap junctions are very special, and they involve these connexin proteins. And here's the cytoplasm of one cell, and then down here is the cytoplasm of a second cell. These gap junctions allow adjacent cells to pass ions and regulatory molecules through a channel between the two cells. And these connexin proteins form this channel and allow molecules to be shared from the cytoplasm of one cell to the cytoplasm of a neighboring cell. And this is a very unique structure found in some cells of our body. The next I want to talk about is local chemical signaling. So paracrine signaling is local signaling. So a paracrine regulator is a molecule secreted by a cell in a local tissue. That means one tissue or area of the body. It's not going to circulate widely throughout the whole body, but these paracrine regulators secrete molecules that diffuse across the extracellular space to nearby target cells, and this is really for mediating a local response. The next one I want to show you is synaptic signaling, and this is where the nervous system will use a nerve to send an action potential to then release a neurotransmitter that can synapse with another cell in the body. For example, this is a muscle cell, and this nerve is telling the muscle cell to perhaps go and contract. So this really involves neurons secreting a neurotransmitter across the synapse to give a signal to a target cell to do some sort of action. And then on the bottom, we have endocrine signaling. Endocrine signaling is hormonal signaling. And this involves glands that secrete hormones into the bloodstream, and these will be transported throughout the entire body. They have widespread circulation. It's going to be systemic. Now, not all cells in the body may have receptors, but all the cells in the body that have receptors for the hormone can detect it, and they can be very distant from the gland that is secreting the hormone. So this is also another really important type of cell signaling. So how do regulatory molecules affect their target cells? Well, there's two different things we need to consider for the regulators, and we have to consider if the regulator is nonpolar, meaning it's lipid soluble, or is it polar and water soluble. So if the regulator is nonpolar, that means it can freely diffuse across the membrane of our cells and then get inside the cytoplasm and then interact with a receptor that is in the nucleus and interact with a receptor in the cytoplasm and travel to the nucleus, where it can then basically help turn genes on or off. And what we're seeing here is that there's a receptor in the cytoplasm, it travels to the nucleus, and then the nucleus will activate the transcription of genes that are being signaled to be turned on, and of course those genes will be translated to create proteins. So the critical thing to know is that for nonpolar regulators, that the receptor is going to be inside the cell. However, if the regulator is polar or water soluble, that means that the regulator cannot get across the plasma membrane. That means the receptor needs to be in the membrane, and it's usually going to be a transmembrane protein that is going to be the receptor. Since these transmembrane protein receptors are stuck in the membrane and they want to affect gene transcription and protein production, they need secondary messengers. So basically other molecules will be produced by some signaling pathway, and those molecules will then trigger and affect gene expression. So basically in this case, the receptor is not inside the cell, it's in the plasma membrane. And then other molecules are going to be transmitting that information to the nucleus. And sometimes it can involve quite a few factors. So let's take a look at one of these polar regulatory molecules would interact with a receptor that's in the membrane. So here we have a receptor that is not stimulated, meaning it has not detected its regulatory molecule. And this receptor is interacting with these G protein subunits called alpha, beta, and gamma. And when alpha, beta, and gamma are bound to each other, and as well as bound to this receptor, the alpha subunit is binding GDP, guanosine diphosphate. Okay, so then when a regulatory molecule binds the special regulatory molecule that regulates the receptor is activated, what happens is the alpha subunit of the G protein, and the G proteins, by the way, are peripheral membrane proteins. They're on the membrane, and they're touching the membrane in this receptor that's a transmembrane protein. So the alpha subunit is going to pick up the GTP, let go of GDP, and when this happens, the alpha subunit will separate from the beta and gamma subunit. So when alpha is binding GTP, it's the activated state. It separates from the beta and gamma. And then either the alpha protein binding GTP will travel in the membrane to hit another molecule, often an enzyme, that will be making an effector molecule, like from big cyclic AMP, for example, that'll then initiate a signal transduction cascade. Alternatively, the beta and gamma subunit, when they dissociate from the alpha subunit binding GTP, they can activate an effector, and these effector molecules will start a signal transduction cascade where the effector molecules will be released into the cytoplasm. So after they've done their job, the A subunit will will basically hydrolyze the GTP to GDP and phosphate, and once GDP is binding the alpha subunit, it'll reassociate with the beta and gamma subunit and then be bound to the unstimulated receptor. We'll be looking at specific examples of the G protein cycle later, but I just wanted to give you a quick introduction to this G protein cycle, because it is something we'll be talking a lot about in greater detail as the semester goes forward. So I just want to thank you for your attention and wish you a great day. Take care. Thank you.